Now, with the background knowledge of chloroplasts, pigments, chlorophyll molecules, and what type of light is required for photosynthesis, now we are in a position to understand the first phase of the photosynthetic reactions, that is light phase reactions, or they are also known as photochemical reactions. Chemical reactions initiated by light. They are cyclic and non-cyclic photophosphorylations. Emerson demonstrates the existence of two photosystems. Emerson demonstrates the existence of two photosystems. Photosystem 1, photosystem 2. How, on what basis he was able to divide the photosystems into 1 and 2. The sudden fall in photosynthetic yield in the far red region greater than 680. When green plants or chloroplasts are exposed to the light of more than 680 nanometers, he observed that the photosynthetic rate suddenly fell down, decreased, compared to red region of the electromagnetic spectrum less than 680. This phenomena he observed. Again, the sudden fall in the photosynthetic in the far red region more than 680, 700 or above 700. So then he analyzed why this sudden fall. This sudden fall phenomena is known as Emerson's Red Drop. Emerson's Red Drop. Emerson's Enhancement Effect, followed by Enhancement Effect. The first one is Emerson's Red Drop, fall. Then in another experiment, he exposed the chloroplasts, green plants, for two types of lights simultaneously. More than 680, less than 680. Two types of lights simultaneously. When he simultaneously exposed the light of both wavelengths, more and less of 680, the rate of photosynthesis was far than the sum of the red light and far red light photosynthetic rates. He observed a surprising increase in the rate of photosynthesis when both of lights are exposed to chloroplasts. Suppose 2 plus 2 not 4, but here 2 plus 2 6. Higher than the sum of the red light and far red light. This is known as Emerson's Enhancement Effect. And this led him to propose the concept of existence of two photosystems. So there should be one photosystem absorbing the light less than 680 and another system more than 680, near 700. So he came to this type of conclusion. He proposed the photosystem 1, photosystem 2. Emerson 1943 concluded that there must be two different photosystems involved in photosynthesis. One driven by short wavelength light, PS1, another PS2, long wavelength light. But they work together to enhance the efficiency and convert the light energy form into chemical energy absorbed by the plant. So there must be two photosystems. Photosystem 1, the short wavelength, photosystem 2, long wavelength. So these two photosystems function simultaneously because the light will be falling on both the systems. When they work together, the photosynthetic rate increases. Light reactions or photochemical reactions, these are light dependent because energy is required. That energy comes from the light, therefore they are light dependent. Occur in grana of chloroplasts. These reactions include light absorption, next water splitting or photolysis of water. Because of that, release of oxygen, formation of high energy intermediates, temporary compounds, ATP, it is energy carrier, and other NEADPH, hydrogen or proton carrier. So the net result in photochemical reactions or light dependent reactions, one is light absorption, second splitting of water, photolysis of water, third release of oxygen, fourth formation of high energy chemical intermediates ATP and NADPH2. So but chemical reaction was what is the first reaction in photosynthesis is this, water splitting. When the light falls on the chlorophyll, automatically it is incident, it is absorbed. But the first reaction is this. Then light absorption. Light is absorbed by the pigments, chlorophylls, carotenoids. Pigments are organized into discrete photochemical reaction centers known as photosystems. What are photosystems? The chlorophyll molecules, carotenoid molecules are not randomly distributed. They are well organized to trap the energy, to absorb the light, to channelize the energy so that light energy is converted to chemical energy. They are organized into discrete photochemical reactions known as photosystems. Two types of photosystem, photosystem 1, photosystem. The photosystem, whether it is 1 or 2, consists of two closely linked, functionally, structurally linked components, an antenna complex surrounding the reaction center in the center. I will show you the diagram. It consists of hundreds of pigment molecules surrounding hundreds of pigment molecules that harvest, that absorb and direct the energy, captured light energy, and channel to reaction center. Then reaction center, which is present in the center of the photosystem, reaction center surrounding that antenna complex. The reaction center consists of one or more chlorophyll A molecule in matrix of protein that passes the energy out of the photosystem. So what this? It collects the energy and transfers the energy to other molecules, just like plastocyan and plastoquinone. Chlorophyll in matrix of the protein, and that passes the energy out of photosystem because it will be in excited state. In excited state, it loses the electrons and that pass to the other electron acceptors. So this is the photosystem structure diagrammatically. The antenna molecules surrounding all these are antenna molecules, different chlorophyll, carotenoid, all these molecules. This is a reaction center. which consists of chlorophyll A. When light in the form of photons, discrete units, fall on these molecules, the energy will be transferred from one to ultimately to the reaction center. The photons fall on this, and all of them collect that energy and ultimately transfer to the reaction center. Now this reaction center, wherein the chlorophyll A molecule is present, that a chlorophyll A molecule becomes excited, loses the electrons. In excited state, it loses the electrons. That electrons will be accepted by some other, we will see later, electron acceptor. These are chlorophyll molecules. So this is the structure of a typical photosystem. Reaction center, antenna molecules. Antenna molecules contain different types of chlorophyll molecules, carotenes, xanthophylls, but the reaction center consists of chlorophyll A predominantly, and all the energy harvested, collected by the antenna molecules is ultimately directed to the reaction center. The antenna complex captures the photons, sunlight. Is a web of chlorophyll molecules linked together and held tightly on the thylakoid membrane by matrix of the protein. This complex, antenna complex, also consists of carotenoid pigments, carotene and xanthophyll. The reaction center, just go to this reaction center, is a transmembrane pigment protein complex, chlorophyll A, and again some protein molecules. One or two chlorophyll A pigments passes an excited electron to acceptor molecule quinone. So when it collects the energy from antenna molecules, surrounding molecules, so it becomes energy, it becomes excited. In the excited state, it tends to lose the electrons. So those electrons are accepted by quinone, reducing and converting to a strong electron donor. This quinone, initially in the oxidized state, upon accepting the electrons, it becomes reduced. Again, it is a strong electron. It can donate the electrons to other electron acceptors. Photophosphorylation. What is phosphorylation? Synthesis of ATP coupling the ADP and phosphate, inorganic phosphate, is known as ATP. Synthesis of ATP, how? By coupling ADP, diphosphate, and inorganic phosphate, is known as phosphor. So we know there are three types of phosphorylations: photophosphorylation, oxidative phosphorylation, substrate level phosphorylation. But here the type of phosphorylation is induced by the light, therefore it is called photophosphorylation. It is an endergonic process, energy requiring process. This is energy rich, energy less. So therefore it requires the input of energy. Synthesis of ATP driven by the light, because the energy required for this process comes from the light, therefore known as photophosphorylation. Two types of phosphorylation: cyclic photophosphorylation, non-cyclic photophosphorylation. Cyclic, non-cyclic. Then according to what is the difference, main difference? Cyclic, non-cyclic. Cyclic means like this. Non-cyclic means not following the cyclic pathway. According to the destiny excited electrons, two types of cyclic photophosphorylation. In this, the electrons coming out of the photosystems return to the same photosystem. passing through different electron carriers. That is cyclic. And during this electron transfer, energy released is utilized to pump the protons from stroma to granulum and build up the proton motive force or proton gradient that is required for ATP synthesis. The PMF is proton motive force is utilized to synthesize ATP through an enzyme called ATP synthase. So this is cyclic photophosphorylation. So this is photosystem 2, 680. Photon light becomes excited. So imagine here all the antenna molecules. This is there will be an amine manganese dependent enzyme. Upon absorbing the light, getting the light from the surrounding molecules, it becomes excited. In the excited state, the electrons are emitted out. These go to an electron acceptor, excited reaction center. These also become excited, and ultimately these electrons come back to the same photosystem. This is called cyclic pathway of electrons. During this process, what happens? The proton gradient is established. Because of this, water is split, 2 protons, 2 electrons. These electrons are accepted by NADH and proton motive force is created. So this is cyclic photophosphorylation. Electrons return to the same photosystem. Now, this cyclic photophosphorylation, remember, not present in higher plants. It is reported only in bacteria, green purple bacteria, sulfur bacteria, like that. But in higher plants, there will be 2 photosystems, PS2, PS1, one dependent on another. This is called Z scheme. Just like Z, inverted Z. Look at here, this is a PS2 system with the reaction center P680. Here, in one way, it is called non-cyclic. The electrons pass through excited state, the chlorophyll molecule excited, pheophytin Q, quinone, plastoquinone, cytochrome B6, ferrous complex, cytochrome F, plastocyanin. Ultimately, those electrons are accepted by PS1 with the P700. Simultaneously, light will be falling on this also. It is accepting the electrons from this system, and light is also again, these chlorophyll molecules become excited, lose the electrons, and these electrons pass through pyridoxines, then pyridine nucleotide, ultimately accepted by NADP. It becomes NADPH by accepting the electrons. Sometimes the electrons may come back this. In that case, it will be known as cyclic photophosphorylation. So, what you need to understand here, the cyclic and non-cyclic photophosphorylations, though explained differently, but function wise they are one dependent on another. This is dependent on this, this is dependent on this. When the electrons coming out of this are not accepted by this is excited state has no use. So one dependent on another. So it is called the Z scheme. In higher plants and cyanobacteria, this type of light phase reactions will be taking place. So you cannot say here whether cyclic, non-cyclic. In fact, it is a non-cyclic photophosphorylation, reaction center 2, reaction center 1, and this is light reaction, green plants and Z scheme accepted by this. Non-cyclic photophosphorylation, the electrons released do not come back. In cyclic they come back, but in non-cyclic do not come back and instead go to another pigment system. The deficiency of electrons will be fulfilled by the electrons coming from splitting of water. So look at here. So this loses the electrons, and those electrons are accepted by P700. Automatically this system falls short of electrons, and this shortage is fulfilled by the electrons coming from the splitting of water, photolysis of water. And here, for this system, suppose if it loses the electrons, that deficiency is fulfilled by the electrons coming from this side. Another pigment system, the deficiency of electrons will be fulfilled by the electrons coming from splitting of water. In non-cyclic photophosphorylation, both ATP and NADPH are synthesized. Suppose if there is cyclic photophosphorylation, but I emphasize again, in higher plants and cyanobacteria, photosystem 1, photosystem cyclic, non-cyclic, one dependent on other, ATP and NADP both are synthesized. But in bacteria, certain bacteria, there will be cyclic phosphorylation returned to the same photosystem. In that only ATP will be synthesized. Now, cyclic photophosphorylation, non-cyclic photo. PS1 involved, both PS1, PS2. Reaction center 700, P6CIT. Electrons released or returned back, they do not return. Photolysis of water, does not take place, but there is photolysis of water. Only ATP synthesized, both ATP and NADPH. This is energy carrier, temporary energy carrier. This is proton and electron carrier, reducing power. Together, these two together known as assimilatory power, required for dark phase reactions. Phosphorylation takes place at two places, but only at one place. It doesn't require an external electron donor, requires electron donor like H2O or H2S or NADH. It is not sensitive to dichloromethylurea, it is sensitive to dimethylurea inhibited. So that is the end of light phase reactions. In the next class, we will consider how the ATPs are synthesized. What is the mechanism involved in phosphorylation? Strictly speaking, photophosphorylation. We have understood that in cyclic photophosphorylation, ATPs are at two places, in non-cyclic at one place, and this is called, synthesis of ATP is called the phosphorylation, photophosphor. But what is the mechanism involved? That mechanism is explained through chemiosmotic theory that we will understand in the next lecture.